Yung Tubbataha kasi, we noticed recently na dumadami yung microalgae primarily. Dumadami rin in some places yung mga sponge. In some places, dumadami yung soft coral. In other places, isang klase ng cyanobacteria. So, yung hypothesis namin is those increases are driven by a change in the nutrient levels. So, parang napapalitan yung mga coral ng mga ibang organismo na normally naman nakikita sa reef, pero yung disturbing for us is dumadami sila at kumukonti yung coral. Ang hypothesis namin ay dahil dumadami yung nutrients sa lagoon, at ang isang possible source ay yung mga ipot ng mga seabirds.
Magandang araw mga kapwa-siyentista at welcome sa Behind the Science podcast kung saan kakausapin natin ang ating mga scientists para ibahagi hindi lang ang mga resulta ng pag-aaral nila kundi pati na rin ang mga behind the scenes or behind the science stories nito. Simula sa kung paano nila ito naisipan gawin hanggang sa na-publish na nila ito. So our guest for today, I'm sure madami sa inyo ang nakakilala sa kanya. So, Dr. Wilfredo Licuanan or Sir Al Licuanan is a distinguished full professor and a university fellow at De La Salle University. He is also a fellow of the California Academy of Sciences. He is a recipient of the 2016 CHED Republica Award, the 2018 Environmental Science Award of NAST, the 2013, 2017, and 2020 St. John Baptist De La Salle Pillar of Excellence Award of DLSU, the 2022 Force of Nature Award of WWF Philippines, and the 2021 and 2024 Outstanding Book Award of the National Academy of Science and Technology. His grade school to master's degrees came from the University of the Philippines, Diliman, and his PhD in biology is from the University of Southern California. Sir Al Licuanan is currently with the Department of Biology and is the founder of the DLSU Brother Alfred Shields Ocean Research Center or Shore Center. He has led research teams to many previously unstudied reefs around the country, including the Kalayaan Islands, where he discovered a new coral species, Leptoceris kalayaanensis, and the Pacific Ocean coasts from Luzon to Mindanao. He led a nationwide reassessment of the status of reefs around the Philippines, and his current focus is setting up a national reef monitoring system and a threatened coral species mapping effort. So without further ado, let's all welcome Dr. Wilfredo Licuanan. So welcome po sa podcast, Sir Al.
Thank you, Paul.
Ayun. So for today, ang episode natin is about Tubbataha Reef. And para sa mga first-time viewers, i-encourage ko lang kayo na i-check din yung previous episodes natin. So particularly yung episode 40 with Mr. Gabriel Hildawa. So si Gab ay RA ni Sir Al. Tama po ba? Di ba, sir?
Yes,
And then
yes.
episode 53, paki-check nyo rin with Mr. Jeffrey Munar. So ang pinag-usapan namin doon ay yung sea level history from 18,000 years ago up to present, which was recorded by the morphology or yung shape ng Tubbataha Reef. And ngayon, ang pag-usapan natin ay ang present day na status ng Tubbataha Reef with Sir Al. And as usual, bago natin yan puntahan, alamin muna natin yung origin stories ng ating mga guests. So Sir Al, first question po namin palagi, kailan nyo po na-realize na gusto niyo maging isang scientist?
Well, yung bata ako I spent a lot of time reading because my mother was a librarian sa College of Veterinary Medicine, and most of the time I was looking at maps. And so I enjoy looking at maps, I enjoy geography, and so along with that came the love to travel. So, pagdating sa college, pinili ko yung course na naisip ko madaming field trip. So, there was a program in UP Diliman, PS Marine Science. So, I signed up for that.
BS?
Back then,
Marine Science, sir?
yeah, yes, we were the last batch. Actually, there was a batch after us, pero walang nakagraduate from that batch. Nag-shift out silang lahat. Ako, I shifted out sa third year because hindi na available yung ibang courses na kailangan namin.
Ngayon ko lang nalaman na may BS Marine Science pala na na course dati.
Yes, it used to be under the Department of Zoology, which as you know merged with botany to become the present Institute of Biology.
Ano pa yung backstory po pala nung yung species ng coral na na-discover nyo? How did that happen?
Well, we were part of yung first series of expeditions to the Spratlys, and I chanced upon Leptoceris calayanensis. It was along a wall, about 40, 50 feet deep. It was very distinctive. Yung genus kasi na yun, normally hindi nagkaka-spines, but this one had spines. Kaya some of my students then called it the durian coral because mukha talaga siyang durian in the sense ng mga maliit na spine. Maliit na spine is about 2-3 centimeters.
Yung process po ng pag-identify ng species na yan, paano po siya nangyayari? How do you define na bago talaga to? Or may database po ba yan na global database or something?
What? Ngayon, yes. There's a site, Corals of the World org. And so that's one of the main sources for identifying corals in the field. Kasi may pictures siya ng both yung skeleton saka yung live na sample. Ang mga corals kasi kailangan mo ng sample, kailangan mo ng skeleton to identify the key features that distinguish species from each other. So, like I said, medyo distinctive siya for its genus. And so it was obvious right away na it was a new species because medyo kaiba siya din sa ibang mga kasama niya sa genus Leptoceris.
Ay, dyan pala sir, curious ako kasi 'di ba kapag sinabing coral, yung lang yun 'di ba sir, yung parang maliit na organism. Tapos yung nakikita natin yung parang skeleton niya na. So kapag sinabing coral, yung maliit lang na yun or pati yung buong skeleton?
Yung coral is the organism. Yung skeleton ng isang colony ay tinatawag na corallium. Yung sinasabi mo na maliit na animal is dahil yung coral ay binubuo ng mga maliliit na modules, ang tawag sa kanila polyp. One way to think about polyps is para siyang mga dahon ng isang puno. So, ang isang coral ay madaming polyps.
So, kapag sinabi pong bagong species siya, yung ibig sabihin ba nun, iba yung itsura nung polyp or yung corallium na yan? Or both siya na iba talaga siya?
For most species, yung skeleton rather than yung polyp. Merong ilang species na kailangan mo makita ng buhay bago mo ma-distinguish yung species. Pero generally, ang coral taxonomy, ang pag-identify ng corals ay nakabase sa skeleton. Part of the reason for that is dahil ang mga pioneers sa pag-identify ng mga corals ay yung mga nag-aaral ng mga fossils. And since skeleton lang ang natitira sa mga fossils, sa mga fossil reefs, yung naging basis ng pag-identify yung skeleton.
I see. And then over the years, sir, nakailang dives na kayo and ano yung mga paborito nyong puntahan na dive sites?
I actually have lost count because I don't log dives anymore. Even want to try to estimate kung nakailang dives na ako. I've been diving since about 1985, 86. So, hindi ko pinapanganak most people in the audience likely. So, matagal na ako nag-dive. Tubbataha is one of my favorite places. And except for a few years during the pandemic, I've been going to Tubbataha for over 12 years.
Outside yung mga sikat na diving sites or yung parang mga unexplored na sites, ano pa yung sa tingin nyo yung magandang lugar na hindi masyadong napupuntahan ng ibang mga tao?
Yung Tubbataha is one of them, pero yung municipality ng Cagayan and Tubbataha is part of Cagayan, has other islands and reefs that are as beautiful. They are not as large, but they can be quite distinct. Yung may advantage kasi yung mga reefs na nasa gitna ng karagatan, like nasa gitna ng Sulu Sea, Cagayan at ang Tubbataha. Dahil malinaw yung tubig, and dahil malinaw yung tubig, wala masyadong sediments, that sort of thing. Medyo distinct ang mga coral assemblages, yung mga areas na madaming coral. Yung mga dumadami ng species, hindi normally dumadami sa mga fringing reefs, yung mga reefs na nasa tabi ng mga isla. So yun yung dahilan kung bakit very interesting at very scenic ang mga reefs sa gitna ng karagatan.
Ayun. So since nag-uusap naman na tayo ng coral reefs, siguro punta na natin yung study ni Sir Al. So ang title nito ay Recent Changes in Benthic Cover in Tubbataha Reefs Natural Park, Sulu, the Philippines. So sir, can you explain muna ano yung paper nyo in one sentence?
Yung Tubbataha kasi, we noticed recently na dumadami yung ilang uri ng tanim, microalgae primarily. Dumadami rin in some places yung mga sponge. In some places, dumadami yung soft coral. And in other places, isang klase ng cyanobacteria. So, yung hypothesis namin is those increases are driven by a change in the nutrient levels. So, parang napapalitan yung mga coral ng mga ibang organismo na normally naman nakikita sa reef, pero yung disturbing for us is dumadami sila at kumukonti yung coral. And so yun yung sinubukan namin intindihin.
Ano po yung possible cause po nung ganong change?
Yung hypothesis namin is may kinalaman sa pagdami ng nutrients sa lagoon, particularly sa South Atoll ng Tubbataha. Kasi may North Atoll, may South Atoll, at merong Jessibis Reef na they're separate reef structure. Mas noticeable yung changes na describe ko sa South Atoll. And ang hypothesis namin ay dahil dumadami yung nutrients sa lagoon, at ang isang possible source ng nutrients ay yung mga ipot ng mga seabirds. So, in-examine namin yung possibility na yung pagbabago sa reef ay may kinalaman sa pagdami ng seabirds na nagne-nest dun sa mga islets.
Ay, dito po pala sir sa study nyo kasi sinabi nyo recent change so 2018 to 2022. Prior to this study, may similar assessments na po ba na nagawa sa sa Tubbataha?
Yes, yung paper na yon na kalalabas lang sa Philippine Journal of Science, merong earlier na paper, co-author ay ako saka si Sandra Benthic. And doon namin una pinropose na may kinalaman siguro yung changes Tubbataha doon sa increase ng nutrients. So dito sa bagong paper, we tried to validate that masinsin yung analysis dahil dun sa unang paper, we just classified yung mga microalgae as algal assemblage, yung term na ginamit namin. In this case, sinubukan namin i-distinguish yung cyanobacteria, turf algae, and tinignan na rin namin yung changes sa dami ng sponges at soft corals.
Kanina sir, nabanggit nyo na yung hypothesis nyo sa change ng benthic cover ay dahil sa nutrient input. So curious po ako doon na, for example, i-take out natin yung nutrient input sa equation. Naturally po ba yung coral reefs nag-undergo sila ng change in benthic cover or wala?
That's a very good question. Depende kung ano yung environmental conditions dun sa area where the coral reef is. For example, kung yung isang reef nagkataon na napuntahan mo when kadadaan lang ng isang bagyo, the nature of the reef, kung ano yung madaming organism, ano yung konti, will be different before the typhoon and after a typhoon, especially kung tinamaan talaga siya ng isang malakas na bagyo. So, kung may isang reef na tinamaan ng bagyo, may mga corals na matatanggal, merong madami ng mababasag, and typically, ang mga unang dadapo na corals doon ay yung mga corals na buwanan mag-reproduce as opposed to yung corals na once or twice a year lang mag-reproduce. Magandang halimbawa dyan yung kung saan nasadsad yung USS Guardian, yung US warship sa Tubbataha. Because minonitor din namin yon since 2013 when the grounding happened. Sa una, natanggal halos lahat ng coral in a small corner ng reef. And may mga fragments ng coral na nabuhay dahil nagkataon na sa mga depression sila, mga butas. Sa side sila ng mga butas na yon. Pero yung una mong makikita na darating in the first 4 years or so ay isang klaseng coral na mabilis tumubo, madalas mag-reproduce, but also medyo fragile sila at sensitive sa pag-init ng dagat and so on. So, anyway, ang ibig sabihin nun is meron talagang predictable changes yan, yung tinatawag na ecological succession. Kung sa lupa, ang unang dadapo ay mga damo at mga ilang weeds, and then later pa papasok yung mga shrubs at mga puno. Meron ding equivalent na ganun sa coral reef.
Pero yung ganong process po, how fast does it occur? Like within a year ba nagre-recover yung reef or more than a year?
No. It takes a long time. For example, may mga reefs din kami na monitor using the same methods sa Batangas at ibang bahagi ng Palawan. Merong isang reef dito sa Western Batangas, sa bayan ng Lian, na noong 2010 dahil sa pag-init ng karagatan, namatay kalahati ng corals, and yung recovery took 7 to 8 years. Sa Palawan, ganun din na-affect ng bleaching, same year, 2010. Pero in one reef, the recovery happened in 4 years. So 4 is fast, 7 is normal, pero unfortunately, madaming reefs na hindi nagre-recover. So, doon sa parehong bayan na sinundan namin yung reef na nag-recover in 4 years, may mga reefs, mga 10 kilometers away na 9 years later, wala pang pagbabago since namatay yung corals in 2010. So, it's a slow process, and certain events or activities prevent recovery.
Kanina sir, nabanggit nyo na yung backstory ng study na to ay in a previous study na ginawa nyo, napansin nyo na yung benthic cover na change. So siguro yung question ko doon is, paano nyo po nasasabi na yung may ganong change na nangyayari? May specific spots lang po ba kayo na pinupuntahan over time, or parang iniikot nyo yung buong reef para masabi na may napansin kayo na change?
Okay. Usually, and nasa guidelines to, for example, ng Department of Environment and Natural Resources, meron kang monitoring at meron kang assessment. Ang objective mo sa monitoring is to detect even small changes. So you want the monitoring to be sensitive to the changes that you wish to measure. But in order to do that, that means meron kang sinusundan na maliit na area over time. Okay? So, yun ang gamit ng monitoring. Ang gamit naman ng assessment ay the assessment allows you to make statements about ano nangyayari dun sa outside dun sa mga areas na minomonitor mo. So, yung una mong tanong, paano namin napansin na nagbabago yung meron kaming monitoring na ginagawa namin sa isang area na in theory ay 75 by 25 meters in dimension. Nasa mababaw na bahagi siya ng reef, between 6 meters and 2 meters depth. It's about the size of 3 to 4 basketball courts.
Mm-hmm.
Tapos, sinasample namin yung area na yun. Yung sampling means naglalagay kami ng measuring guide in random places within that 75 by 25 meter station. We call it a monitoring station. Within the monitoring station, meron ding mas maliit na area. It's a 4 by 4 meter plot na minamark talaga namin yung corners with metal pegs. And yun is an even more sensitive monitoring dahil kinukunan mo ng madaming litrato yung isang napakaliit na area. And then, gumagawa ka ng mosaic ng image na yun. And pwede mo talagang ipakita kung lumaki ba ang isang specific na coral, lumiit, or namatay, or kung nadagdagan. That's the same method that we use to monitor yung ship grounding, katulad ng binanggit ko kanina. We also monitor yung pinagsadsadan ng isang US warship at ng isang Chinese na fishing boat.
Pero yung pagpili po nung monitoring station na yun na ilan ba yun? 75 by 25 meters. Paano nyo po yun pinipili, and how do you argue na etong lugar na to, yung makikita natin dito, it would be representative of the entire reef?
Okay. Usually and originally, yung mga minomonitor namin na nagkaroon kami ng reconnaissance surveys yung tinatawag na manta toes, which means may isang tao na hinihila behind the boat holding what's called a manta board. It's just a piece of plywood that allows you to kind of steer kung nasaan ka relative to the boat that's pulling you. Based on that, you can choose representative sites na itong area na ito, dahil madami halimbawa ang branching coral dito, then we should have a monitoring station here that represents this part of the reef na madaming branching coral as opposed to another area where the corals are more hemispherical, ganun. That's one way to do it. The other way with the newer rules is gagawa ka ng general assessment. And ang general assessment, ang sampling mo ay random. So, ang guidance na pinaprovide namin is i-divide mo yung isang reef into at least 3 parts. Yung dalawang parts are yung nakatapat sa amihan at sa habagat, the 2 monsoon winds. At yung pangatlong part ay yung hindi nakaharap sa amihan or habagat. And then, you use those strata to define, okay, within this stratum, random ang sampling. Within this stratum, random ang sampling. And kung mas malaki yung isang stratum, mas madaming samples ay kinukuha doon. Based on that stratified random sampling, pwede kang pumili ng mga stations na gagamitin mo for future monitoring.
Ayun, so speaking of Mantato, para pala sa mga first-time viewers dito, please also check yung episode 5 namin with Dr. Maricor Soriano. So sa episode na yun, nag-discuss siya ng technique to rapidly map coral reefs using underwater cameras. So please check it out. So going back dito sa sa discussion natin with Sir Al. So ngayon sir, with that method, paano nyo ngayon vinalidate or tinest yung hypothesis nyo na yung benthic cover ay yung change ng benthic cover ay dahil sa nutrient inputs? Ano yung methodology to test that hypothesis?
So yung describe ko kanina how we sample a monitoring station. Yung monitoring stations namin sa Tubbataha and few other places, lagi silang magkapares. Merong dalawang magkalapit na monitoring station and a pair of stations we call site. So 2 stations, 75 by 25 meters per station. If they are within a few hundred meters of each other, we call a pair. Sa Tubbataha, there are 2 sites in the North Atoll. There are 2 sites in the South Atoll. And like I said, each site is made up of a pair of stations. The reason ginagawa namin yun is dahil, for example, ang dahilan kung bakit kumukonti yung coral mo ay ang pag-init ng karagatan. Dahil ang isang thermal or warming event, kung mangyari, ay nangyayari siya sa malaking area. You would expect na hindi iba ang mangyayari sa dalawang station sa isang site. In fact, hindi rin magbabago yung mangyayari sa isang site at yung another site within the same atoll. Kung uminit ang karagatan, you would expect all the stations and all the sites in Tubbataha to show an effect. Pero, kung ang source ng disturbance mo ay very local, halimbawa, merong sumadsad na floating log, isang troso, or isang malaking float galing sa payaw, it's a fish aggregating device. Kung sumadsad yon sa reef at sinira niya yung corals, hindi mo makikita yung effect sa dalawang stations ng isang site, makikita mo lang siya likely sa isang station.
Mhm.
So, kaya yung dinescribe ko na dalawang station, isang site, dalawang site sa isang atoll, tapos merong dalawang atoll, and yung dalawang atoll, we call that a location. Kaya ginagawa yung tinatawag na hierarchical sampling is para magkaroon kami ng idea kung ano yung dahilan ng pagbabago na nakikita namin.
Mhm.
So, to test our hypothesis about eutrophication, kinumpare namin yung nangyayari sa mga stations, kinumpare namin yung nangyayari sa mga sites, kinumpare namin yung nangyayari sa mga atolls. And ang expectation namin ay dahil mas maliit yung South Atoll at mas maliit yung lagoon niya, therefore, mas mabilis magbabago yung nutrient level.
Mhm.
In-examine namin yung possibility na yung pagbabago ay mas obvious dun sa mga stations at sites sa South Atoll kaysa sa North Atoll.
Mhm.
So, in effect, hindi lang namin tinitingnan kung kaano kalaki yung pagbabago. Tinitingnan namin kung yung pagbabago ay may kasabay dun sa mga ibang lugar na minimonitor namin. At kung yung mga pagbabago na yun ay yung ma-expect mo kung dumadami nga yung nutrients or pataba dun sa tubig. So that's how the research was designed. It was designed to test for yung expectation namin na kung ano dapat ang mangyayari kung ang pag-increase ba ng nutrients ang dahilan dun sa pagbabago.
Then with that, sir, so ano yung na-observe nyo? Is there a statistical ano ba tawag doon? Parang statistical significance dun sa sa change dahil sa nutrient input?
Yes. So we did statistical tests and we showed changes. There are statistically significant results. But yung mga significant results ay hindi lagi nagko-correspond dun sa expectation namin. In-expect namin halimbawa na dadami yung sponges, pero hindi. In some places, yes. In some other places, no. So, hindi siya consistent, which means hindi namin ma-conclude dahil yung findings ay mixed. So, that's one of the results. The other thing we did beyond yung statistical testing is we also did power analysis for the test. Kasi, pag ikaw ay nag-statistics, halimbawa, gumawa ka ng isang t-test, pair mo ang dalawang bagay using a t-test. And yung result ng t-test ay wala kayong na-detect na significant result. When that happens, there's actually two possible reasons why hindi statistically significant yung result ng t-test. The first reason is talagang walang kaibahan yung kino-compare mo with the t-test. And that's usually the conclusion of most people interpreting a t-test. But there is a second explanation, and that's where power analysis comes in. The second explanation is your data is not sufficient to support, to reject the null hypothesis. In other words, masyadong variable or masyadong kaunti yung data mo na hindi mo naman talaga marireject yung null hypothesis na yun kahit na dapat siyang i-reject. So that's what the power analysis does. It allows you to check the non-significant result and the non-significant results kung meron bang power yung inference na. In other words, talaga bang
Mm.
walang kaibahan, uh, kulang lang yung data mo or
Mm.
masyadong mataas yung iyong. And that's what we found na in addition to the results not being completely consistent with our expectation, we also found that many of the tests did not have sufficient statistical power. So in other words, to summarize our work, we showed na we cannot eliminate the possibility na dumadami nga yung nutrients kaya nagbabago yung reef. But it also cannot conclude na the changes are because of an increase in nutrients.
Mm.
And so we recommended na magdagdag pa ng measurements, i-continue yung monitoring. We also recommended na kailangan natin tingnan na merong isang other factor that's causing changes. And that is yung impact ng mga bagyo.
Pero sir, kanina nabanggit niyo po na yung source ng nutrient ay yung ipot ng mga ibon. Paano po kayo nag-arrive sa ganong hypothesis? Ano pa po ba yung other sources of nutrients?
Actually, there's another source. If you talk to the oceanographers, especially at MSI, they've been studying this phenomenon called internal waves. An internal wave, and best to talk to them about this because it is very interesting. If you have a body of water like Sulu Sea, and meron siyang thermal stratification, may isang layer na iba ang density than another layer below it. Pwede kang magkaroon ng alon na nagta-travel underwater at the interface between those two layers. And there's internal waves being generated in the Sulu archipelago that travels across the Sulu Sea, hits Tubbataha, and goes beyond. And yung isang possibility is that event, whenever that happens, and it happens a lot, that event might be bringing cold, nutrient-rich waters from the deep into the shallow areas of Tubbataha. So that's another source of nutrients that must be investigated. But based on a few data points, and I have to emphasize a few data points, parang yung changes na nakikita namin ay nasa maling side ng Tubbataha, kung saan tumatama una yung wave. Okay. So that's why kailangan pa ng additional studies yon. Pero at this stage, we cannot really conclude na yun yung nag-drive. Okay. We also, there are nutrient measurements around Tubbataha. The data points are also very few, and so we cannot really conclude na clearly mas mataas ang nutrient levels sa Lagoon ng South Atoll, that sort of thing. The data is not strong enough to allow us to make that statement. So all we can do as reef scientists is try to understand what's happening to the organisms that we know best. But clearly, other people in other disciplines can help us understand what's going on.
Ayun. So I guess madami na kaming napag-usapan ni Sir Al regarding dito sa paper, and I think punta naman natin yung more on behind the scenes ng study. So yung study po na to, ano po yung naging funding source niya?
Our funding, yung immediate around that paper is the OSP Picard. the reason I say immediate is because we've been monitoring Tubbataha for over a decade.
Mhm.
And, but the grant na binanggit ko, that was a 2-year grant about the time of the pandemic. So yun yung in-acknowledge namin na support for that particular paper dahil yung project na yun ang nagpasahod din sa karamihan ng authors.
And so in this study po, ano yung mako-consider nyo na challenge na na-experience nyo in doing this?
Well, I mentioned kanina na the grant was given about the time of the pandemic, which meant we couldn't go to Tubbataha. Then that was one of the bigger challenges. But what we did naman talaga, the funding was given about the time of the height of the pandemic. And so what we proposed was to go back to 12 years of pictures of the reef, taken using that methodology we used to sample our monitoring stations. Those are image-based methods. So you can identify corals, you can quantify their abundance from all these pictures that we take.
Ayun. And in this study, sir, at what point niya siya nasabi na i-publish na natin to? Was there a specific figure dun sa paper na nagawa nyo and then nag-decide kayo na i-publish, or?
So we covered 5 years in that analysis, and there was a small overlap between yung period covered in that analysis and the period before that, dun sa earlier paper na nabanggit ko, that also came out in the Philippine Journal of Science. So basically, dinugtungan lang namin yung time series. And we felt na, yun na, why wait? Kung may 5 years of data ka, complementing an even longer data set, we did not hesitate na we had enough data.
Pero ang interesting din, sir, ng ginawa nyo, kasi dito sa paper nyo, parang sinabi nyo na kailangan pa ng more data points para masabing yung statistical power ng data nyo. Yung impression ko kasi kadalasan yung mga tao nagpa-publish sila kapag may nakita silang statistically significant na result na affirming yung initial hypothesis nila. Pero sa inyo, parang sinabi nyo na we can't completely rule out yung ganitong possibility sa limited data points nyo. So I think very interesting yung part.
Yeah, the reason our approach is different is also because very few people do power analysis. Very few people actually doubt the statistics until it's backed up by another level of statistical analysis. So because we did that, that allows us to make conclusive statements about for this, clearly kulang pa ang data kahit na 5 taon siya, that sort of thing. There is another work of ours na merong power analysis, and it was covering 101 monitoring stations from around the country. More people need to do power analysis, especially in the environmental sciences.
Ayun, so almost 1 hour na tayo. I guess kailangan natin i-wrap up yung episode. So sir, ano pa yung ma-advise nyo sa mga aspiring researchers?
For my discipline, but many other related disciplines, people have to start early in accumulating a good time series. Okay? We have far too many studies na ang liit-liit ng period being covered. Imagine a curve na a unimodal curve. Okay? May curve na part ng curve pataas, part ng curve level, part ng curve pababa. If you're studying something and yung ganun ang pattern ng halimbawa ng parameter na sinusundan mo, that means na if you're only looking at a small window of time, you might see an increase over time, you might see no change over time, or you might see a decrease over time. And they could be happening to the same parameter in the same place. Hindi mo ma-realize yun unless you have a really long time series. So, we have in the reefs near our marine station, our monitoring data set is already 15 years old. And the sort of statements we can make with a 15-year-old data set is much more conclusive than if you have a 1 or 2-year study. Especially for, like we discussed at the start, an ecosystem like coral reefs, unless you have a catastrophic event, the changes are very slow. So my advice to young scientists is study something kahit na on the side, but accumulate a long time series. Because the understanding that allows is there is no other way. There are a few other ways to can make statements as conclusive as you can with the long time series.
Ayun. And then, last question po, sir. Do you have anything to promote?
Well, we are promoting citizen science monitoring of coral reefs for the same reasons na nasabi ko na. We have to have more time series for more places. Recently, using methods of citizen science, nag-design kami na isang method na allows people to monitor reefs kahit na wala silang scuba, kahit na wala silang specialized training sa pag-identify ng corals. And recently, we showed na yung 2024, because of the warming, because of the successive typhoons, we have reefs that have been degraded in a matter of months, and they've never been as bad as they are now since the last 15 years. So, because of methods like that, in one town, the mayor recently signed an order regulating fishing and tourism activities in a section of a reef that needs some time to recover last year's events. So my point there is the reason we're promoting citizen science because it allows communities to base their management, their reef, the conservation of their reefs on actual data, not just something you saw on YouTube or in the internet or something.
And I guess nakakatulong din siya, sir, dun sa sinabi niyo na yung importance ng time series kasi hindi naman palagi na kayo yung makakapunta sa site every month.
And the way we set it up, central ang role ng municipal government, but central din yung role ng mga high school. The high school students, kahit na hindi sila pinadala sa reef, are involved in the processing of the images and the data. And so it improves STEM education, it promotes monitoring of the reef, and it provides guidance to communities on what they're doing with regards to coral reefs. That's what we're promoting now.
Ayun, so I guess hanggang dito na lang muna tayo. Maraming salamat po, Sir Al, sa pagpapaunlak ng aming imbitasyon, and maraming salamat sa ating audience sa pakikinig at panonood.
Thank you, Ben. Thank you for having me.
And so, see you everyone sa next episode. Madami pa tayong pag-uusapan na research na ginagawa ng ating mga Pinoy scientists.